Today we will learn the structure of peptidoglycan. I have few questions. The first question is, what is the structure of peptidoglycan? Our learning objective is to learn the structure of peptidoglycan. Structure क्या है? Our second objective is to learn why peptidoglycan makes bacteria versatile. Reason क्या है? Why it makes the bacteria versatile? And third thing is, what we do when we get the bacterial infections, how we get rid of the bacterial infections. You know, it has been the talk of centuries when we have read in the history that there are a number of people who die because of a simple bacterial infection, right? Because at that time penicillin was not there, right? So, what are the target sites where we can target a bacteria? Understand? These are all things we will learn today while understanding the structure of peptidoglycan. बहुत सारे सवालों का जवाब मिल सकता है, लेकिन इन सारे सवालों को आपको बायोकेमिस्ट्री और फार्माकोलॉजी जुड़ती है यहाँ पे. Because when you enter into the pharmacology, you will see the mechanism of action of penicillins, mechanism of action of beta-lactamase, right? Beta-lactamase inhibitors. जब तक आपको पेनिसिलिन का जब तक आपको पेप्टिडोग्लाइकन स्ट्रक्चर नहीं पता होगा, तब तक आप उसका आंसर नहीं दे सकते. So if we want to know more and more about the bacteria, more and more about the antibiotics, we need to learn the basic structure of peptidoglycan. And in this lecture we are going to understand the same. Let us see. Let us start. Peptidoglycan. Peptidoglycan is a heteropolysaccharide. It is a heteropolysaccharide. What do you mean by heteropolysaccharide? It is made up of more than one monomeric unit polysaccharide. Heteropolysaccharide का मतलब है it is made up of more than one type of monomeric unit. But it is a polysaccharide. It is made up of large number of monomeric units. And the two repeating units of the peptidoglycan are N-acetyl muramic acid and N-acetyl glucosamine. There are two repeating units. It is framed by the basic fundamental two, by combining the two basic fundamental, we can say, structures. One is N-acetyl muramic acid and second is N-acetyl glucosamine. The two are joined together by glycosidic bonds, as we do in case of disaccharides, trisaccharides and polysaccharides. First we will understand the structure of NAM and NAC. Let us take this NAG. NAG is N-acetyl glucosamine. For this we need to draw the structure of glucose. You know glucose? Glucose is soluble in pyranosic form, right? This is the pyranosic form, and here we have a question whether this is in beta form or in alpha form. So for all times to remember, it is beta. This is also beta. Beta means, where is OH? On the top, right? This is H. N-acetyl, NHCOCH3. Here we have this group attached. And rest, the structure is same as that of the glucose because it has been derived from the glucose. I am substituting the acetyl group at position number 2. The rest everything is same as that of the glucose because it has been derived from the glucose itself. I can number this. It is 1, 2, 3, 4, 5 and 6. This we can call as N-acetyl glucose Is it clear? And now we want to learn about N-acetylmuramic acid. What is N-acetylmuramic acid? N-acetylmuramic acid, if you want to make, again you have to make the glucose. You have to make the glucose. You try to make it. This is this way. This is O. Okay. I will make a ring. Which type of ring? Shabash. कौन सी रिंग बनानी है? पाइलिनोसिक रिंग. शाबाश. पाइलिनोसिक रिंग बनाने के लिए मुझे यहाँ पे बीटा फॉर्म में ये भी है, तो OH is on the top. Is it clear? Okay. Again I will substitute N-acetyl, that is NHCO, what? CH3. Add position number 3. Add position number 3. You will see some changes. Where is position number 3? 1, 2, and 3. Here, 4, and here it is CH2OH. Okay? This is 5. This is position number 4, position number 5, and position number 6. Now look here. I will make it this way. Here it is CH2OH. CH2OH. Here it is H. And here it is OH. Routinely, here it is H. Here it is H. Now, at position number 2, N-acetylmuramic acid is formed from N-acetyl glucosamine plus lactate plus lactic acid. When we combine lactic acid with N-acetyl glucosamine, we can form what? N-acetylmuramic acid. Now see, the structure of glucosamine is same as we are trying to make for muramic acid. But at position number 3, we are adding lactate group to it. The only difference between the two is that at position, in case of NAM, at position number 3, we'll be adding a lactate group to it. So it means here it is H and here it is O. You know it is O. O ko I will get it down this way. O here. And why don't I make it more close, because suppose this is O here, okay? Here you have a lactate group. Lactate is CH3 CHCOOH. You understand? It means this O is interacting with this group. This O is interacting with this group. This is lactate. But again, it is not a complete N-acetylmuramic acid, because N-acetylmuramic acid has a small peptide chain attached to it. See, you try to understand the basic thing, you will understand the whole structure later on. I am adding one more point to this. What I am telling you? I am telling you, in case of N-acetylmuramic acid, you will find at position number, it is same as that of N-acetyl glucosamine, plus a group of lactate and plus a short stretch of polypeptide. Peptide means protein. We are bringing protein into the sugar. It means it is a type of glycoprotein, right? When we add proteins to the carbohydrates, we call them as glycoprotein. Let us see what is that strip of protein. I can make it more clear. I think it is better to make it more clear so that the structure will come all around in between your eyes. Okay. The image is very good, but a little bit it is confusing me. Okay, this I cannot get. Now see, N-acetylmuramic acid, I will make it more closely, more correctly, so that you will visualize. Now look here. Tell me what I have to write here. NHCOCH3. Here we have the same OH and H. At position number 3 we have O. This is O, and this O is bind with the lactate. It is CH3COOH. Rest everything is same. OH here and CH2OH here. What is this group? Carboxylic group. And what is present in the amino acid? You have an amino group and carboxylic group. Carboxylic group can bind with the amino group of the amino acid. I would say carboxylic group of this lactate will bind with the amine group of amino acid. And here you will find, I can write it, this is C double bond O, okay? There is a short stretch of almost 4 amino acids. How many amino acids? The first amino acid is alanine. The second one is isoglutamate. The third is lysine. The fourth is alanine. Very nice. And you will remember, most of the sugars are suitable in D form. Understand? Most of the sugars exist in D form. But most of the amino acids exist in L form. And here it is L, it is D, this is L. L-alanine and D-alanine. Right? So what is the short stretch of polypeptide? First is alanine, second is isoglutamate, third is lysine, and fourth is alanine. This is a short stretch of polypeptide which is attached to the lactate group, which is in turn attached to the N-acetyl glucose amine. N-acetyl glucose amine. This is the structure. This is beta, this is also beta. Now the linkage is glycosidic linkage between 1 and 4. It is beta 1,4 glycosidic linkage. How I will write this beta 1,4? Here, if I will make the numbering, it is 1, 2, 3, 4, 5 and 6. Alright? Now beta means here between this OH and H, the water will go out. Okay? This OH and H, the water will go out, and we will get what we call as a glycosidic bond formed between the N-acetyl muramic acid and N-acetyl glucosamine in this form. You understand? This is the basic structure, basic framework of peptidoglycan. You clear? Any confusion? Any confusion? This is a clear basic structure of N-acetyl glucosamine and N-acetyl muramic acid, which are joined to each other by beta 1,4 glycosidic bonds. Understand? Shall I rub this? Okay, now let's go. अब देखिए आप क्या कैसा है. अब जब हम देखेंगे, now we will draw the complete structure, three-dimensional structure. You will see peptidoglycan is not a linear chain, but it is a three-dimensional structure. And beyond this bond, there are also other bonds which are involved in the formation of peptidoglycan. Now let us understand. If I will try to draw the structure of peptidoglycan, we have the first thing is N-acetyl glucosamine. Let's make it this way. This is suppose N-acetyl muramic acid. Suppose this is N-acetyl muramic acid. Then we have N-acetyl muramic acid here. Okay, N-acetyl muramic acid here. N-acetyl muramic acid here. I will make N-acetyl glucosamine here. Okay, N-acetyl glucosamine here. N-acetyl muramic acid here. How they are connected to each other? They are connected to each other by they are connected to each other by beta 1,4 glycosidic bonds. This is one chain which is, what is this? What we name it as black? Black is NAG or NAM, whatever you like to say. Okay, NAG, okay, if you are comfortable with NAG. This is NAM, NAM. Huh? This is NAG. This is NAM. NAG and NAG. Any confusion? Right? The connection between the two is provided by glycosidic bonds. Right? For example, this is a glycosidic bond connecting this and this glycosidic bond. Here also, everywhere. This is one chain. This is the first chain. Let us move to the second chain. Again, glycosidic bonds will be everywhere. Third. And these two chains are as such, as of now, they are not connected to each other. Right? What will happen to the stability? Stability will be less. Because peptidoglycan is a 3D structure, we have to connect them all. Now look here. Attach it to N-stagmuramic acid. We have a short stretch of polypeptide chain. And what is this polypeptide chain? The polypeptide chain we can have the polypeptide chain here, like in this form. Attach it to this one. First amino acid, second, and then third and fourth. Okay? Is it clear? Similarly, we can make it this way. 1, 2, 3, 4. Here also 1, 2, 3, 4. Attached to this also we have a short switch of polypeptide chain. 1, 2, 3, and 4. This one also 1, 2, 3, 4. 1. What's wrong with this? There also, you understand? Here also we'll make 1, 2, 3, and 4. Here also we have 1, 2, 3, 4. Can you name it? First one is? Second one is? Isobutyl. Third one is? And fourth one is? Alright. Absolutely right. Once you will understand this, you will come to know so many the questions which we have discussed earlier. We will find the answer of all these questions. But provided we have to understand this. Right? Now, still the two chains are not connected to each other. In the formation of peptidoglycan, this is one chain, second chain, third chain, fourth chain. They are existing, but they are not connected to each other. In the formation of peptidoglycan structure, we will see there is formation of pentaglycine interbridge. A bridge is connected between the two, and the name of the bridge is what? Pentaglycine interbridge, connecting one peptide with the other. Like, let us suppose this is 1, 2, and 3rd and 4th amino acid. Okay, this one is the 4th amino acid. This is 4th amino acid. You connect 3rd amino acid. This is 3rd amino acid. You connect 3rd amino acid of first chain with the 4th amino acid of the second chain. You connect 3rd amino acid of first chain with the 4th amino acid of the second chain. There is formation of a bridge, and this bridge is formed by 5 glycine residues. Who is forming this? 5 glycine residues will come and will connect the 3rd amino acid of one chain with the 4th amino acid of the second chain. It means here we have a glycine 1, 2, 3, 4, and 5. Here again, I can say 3rd with the 5, 3rd with the 4. 1, 2, 3, 4, and 5. Pentaglycemic interbreed. Here I can say 3rd with the 4th, 3rd with the 4th. Pentaglycemic interbreed. 1, 2, 3, 4, and 5. Here I can also join 3rd with the 4th. Pentaglycine interbreed. 1, 2, 3, 4, and 5. I can connect this with this with this. But remember, at one time, Suppose you are connecting this polypeptide chain with this by this pentaglycemic interbreed. It is not going to again form one more pentaglycemic breed. So once you will find, suppose a pentaglycemic breed is established, it is established between the two, there is no formation of another pentaglycemic breed by the same NAM. If NAM is participated, NAM and NAM is participated, it will participate once. Then you have to take another one. Suppose this and this has participated. Suppose this and this has participated. Suppose this and this has participated. Not all. It means what we call it as, we call this phenomenon interbreed formation, pentaglycemic interbreed formation, and this pentaglycemic interbreed forms the peptidoglycan as a stable entity for which we require one important enzyme known as, can anybody say? Transpeptidase. The enzyme required for the formation of pentaglycemic interbreed is known as transpeptidase. Sometimes we call it as penicillin binding protein, PBP. Now we are correlating with pharmacology. You might have heard from most of the time that biochemistry is the central subject which is connecting to all the other subjects, right? And really it is very true. Unless and until you do not clear the concepts of biochemistry, you are not supposed to do good in pharmacology, immunology, or even in gynecology or any other subject. Now this is transpeptidase. Transpeptidase is an enzyme. What it will do? It will be catalyzing the formation of pentaglycin interbreeds. It means it will be forming the peptidoglycan more stable. And where this peptidoglycan is present? My second question is, कहाँ पाया जाता है peptidoglycan? Peptidoglycan is present in the bacteria, but bacteria के किस part में? Cell wall. Is there any difference between difference of peptidoglycan in gram positive and gram negative bacteria? What is the difference? Pentaglycin interbreed is present in gram positive bacteria, while is in gram negative bacteria there is only does not matter. I am telling you peptidoglycan. You are telling me suppose 3 with 4. But in another case you say it may be even these are 4 amino acids, but it may be 5 amino acids as well. There may be repeat chain of one alanine more. That's not my concern. My concern is peptidoglycan is present in the cell wall of both gram positive as well as gram negative bacteria. And the peptidoglycan is meant to provide the stability to the cell wall of bacteria. Do you know why the bacteria, a single unicellular organism having no mitochondria, having no membrane-bound organelles, is a successful candidate in the animal kingdom? How it has turned out to be a successful one? सवाल ये है, एक बैक्टीरिया जिसके पास पूरी अवकात नहीं है, अवकात क्या नहीं है, उसके पास एक प्रोकैरियोट है बैक्टीरिया जिसके पास मेम्ब्रेन बाउंड ऑर्गेनेल्स नहीं है, जो सेल के लिए फंडामेंटल है, एंड एक न्यूक्लियस के बदले न्यूक्लियोइड पे काम चला रहा है, एक छोटा सा प्लाज्मिड है, डेट्स ऑल, एंड थोड़ा सा एंडोप्लाज्मिक रेटिकुलम 70 ऐसे. हाउ इट इज मेकिंग ए सक्सेसफुल कैंडिडेट इन द यूनिवर्स? Yes? It is because of the presence of cell wall. Why arthropods have constituted the major animals in the animal kingdom? It was because of the presence of only one thing, that is exoskeleton. So something makes you more. Why plants are everywhere? Why flora? Why we have more flora than fauna? It is because of the presence of cellulose. So these are the structural components. Because of the presence of a rigid cell wall, because of the presence of a defensive type of cell wall, you can see bacteria is really flourishing in this kingdom. You understand? That is important. Now, for the stability of the cell wall, one of the structures is peptidoglycan, because peptidoglycan makes the cell wall stronger and makes it resistant to get attacked by so many things. Because if we take the cell wall out, you will see anything can come inside the cytoplasm and it is going to destroy the bacteria. Now understand one thing. There's a difference between the cell wall of gram positive and gram negative. What is that difference? I will try to make it somewhere here. Okay. I will try to show you here. You might have read it in microbiology. You have done microbiology part, or it is still with you? माइक्रोबायोलॉजी पढ़ते हो? तो वहां तो पढ़ा होगा ना, पहला ही है ना? So tell me about the envelope of gram positive bacteria. What do you know? What do you know about the envelope of gram positive bacteria? Suppose we have a gram positive bacteria here. I will make this envelope. You will find the cell wall of gram-positive bacteria has a thicker layer of peptidoglycan. This is peptidoglycan. It is a thicker layer of peptidoglycan. And below this peptidoglycan, what you will find? You will find as usual what we call a cell membrane. Cell membrane. Understand? You will find a cell membrane. Cell membrane we have here. For example, here we have a cell membrane in the gram-positive bacteria. It means what is this? Peptidoglycan. And what is this? Cell membrane. And what is inside? It is cytoplasm. Everybody knows. When we talk of this, I have taken this color. When we talk of, we can say gram-negative bacteria. You will find, yes, this peptidoglycan is present. For example, this is the peptidoglycan. But the peptidoglycan is not thick, it is thin. It is thin. And below this peptidoglycan, you have what we call as periplasmic space. Do you remember? What we have? We have a periplasmic space. And below this space you have what we call as plasma membrane. So here we will find a difference. And above this we can say peptidoglycan, you will find there is outer membrane. There is an outer membrane. Probably you may be knowing. There is an outer membrane. Is it clear? Now see, what is this? This is the outer membrane. This is the outer membrane, OUTR, outer membrane. And what is the second layer? It is peptidoglycan. And what is this one? This is known as periplasmic space. It is a space, periplasmic space. Can you tell me what is present in the periplasmic space? Huh? No. Inside this periplasmic space. And this, yeah, very nice. Who said this? Cheers for it. Very nice. Enough. Beta-lactamase enzyme is present in the periplasmic space. You are right. Very good. It is plasma membrane. Now see, let us discuss about the mechanism of action of penicillin. The first antibacterial which was produced is known as penicillin. Do you know this penicillin was a marvelous discovery, wonderful discovery of science? You know it? And this penicillin has a beta-lactam ring. It has a beta-lactam. What is happening? Suppose this penicillin, when we look at the mechanism of action of this penicillin, it acts on transpeptidase. It acts on transpeptidase enzyme, or which we call as penicillin binding protein. What it will act? It will act on the transpeptidase or penicillin binding protein. It means it is going to deactivate this enzyme. It is going to deactivate the transpeptidase enzyme. What will happen? Once the enzyme is deactivated, you will not see the formation of pentaglycemic intermediate. And what will happen? The stability of the peptidoglycan is lost. Once the stability of peptidoglycan is lost, the cell wall of the bacteria is not formed. the bacteria is not in a position to form the cell wall. How we can target the bacterial cells? We can target the bacterial cells by seeing what is present in them which is not present in us. Right? And peptidoglycan, peptidoglycan is not a constituent of any of our cell. So if I will target the peptidoglycan, we can target the bacteria inside me because it can selectively go and act on the peptidoglycan. Do you know the other structure, other possibilities by which we can target the bacterial cell? One is the 70S ribosome, because 70S ribosome is not part of the human eukaryotic cells. So we can target the 70S ribosome and kill the bacteria. One of the reasons of the first antibiotic which was discovered has a mechanism of action by which it is acting on the enzyme known as transpeptidase or penicillin binding protein, and it will be deactivating it will not be forming the pentaglycemic interbridge, hence the cell wall formation is inhibited. But is it true, really true in gram-positive bacteria, in gram-positive and gram-negative bacteria? Yes, it is. There is a little bit of difference. If we talk of Staph aureus is a gram-positive bacteria. Yeah, there is no issue if you have infection of Staph aureus. We can give you simple penicillin and you are completely out of the infection. But when we talk of gram-negative bacteria has a different type of cell wall. Yes, it is going to, the penicillin is going to affect the peptidoglycan of gram-negative bacteria. But just now we just said, this girl said, periplasmic space has beta-lactamase enzyme. And penicillin has a beta-lactam ring. Penicillin has a beta-lactam ring. And the effect of penicillin is because of the presence of beta-lactam ring. Now when beta-lactamase is present in gram-negative bacteria, it can directly attack on the beta-lactam ring of gram-negative bacteria. It can directly act on the beta-lactam ring of penicillin. है ना? ये किस पे अटैक करेगा? पेनिसिलिन के beta-lactam ring पे, and it will deactivate the penicillin. So we can say the gram-negative bacteria are not, they can be resistant to penicillins because of the presence of beta-lactamase enzyme, which is found in the gram-negative bacteria that can directly act on the beta-lactam ring of the penicillin and can deactivate it. So then we have a new class of antibiotics, beta-lactamase inhibitors. New category of antibiotics, beta-lactamase inhibitors. You might have read penicillin, simple. You might have taken ceftriaxone, right? You might have taken cefotraxime. You might have taken ceftiofur. You might have heard the name of tazobactam, salbactam, cloxidine, right? When we talk of this group of drugs, you will see we have salbactam, we have tazobactam. We add. Suppose we have ceftriaxone. Ceftriaxone is a cephalosporin, but this ceftriaxone is not resistant to beta-lactamase. It can be deactivated by beta-lactamase. But when we add salbactam to it, we have a combination. आप अगर कभी clinics गए हो तो आप देखना. You have Vocef plus. Vocef plus means it is what? It is ceftriaxone with salbactam. It is amoxicillin with cloxidine. It is ceftriaxone with tazobactam. It is amoxicillin with tazobactam. आप रोज खाते हो, क्या खाते हो एंटीबायोटिक जिसमें amoxicillin plus tazobactam आता है? हाँ? We call it as augmentin. What is inside the augmentin? Amoxicillin and potassium clavinate. What is this potassium clavinate? Amoxicillin is a cephalosporin, understood. But what is this what we call amoxicillin and potassium clavinate? What is this potassium clavinate? आपने पता नहीं बहुत बार IV लगाया होगा. Ceftriaxone with salbactam. Ceftriaxone is a cephalosporin. What is the salbactam? Ceftriaxone can take care of gram-positive bacteria. But if your infection is gram-negative, then you have to give a beta-lactamase inhibitor. And this beta-lactamase inhibitor is tazobactam, potassium clavinate, or we have salbactam. So therefore, in order to make an antibiotic, a broad spectrum antibiotic, we have to what? We have to give it in a combination. We have to use antibiotic like penicillin, but along with this, we have to ensure the use of the salbactam, tazobactam, potassium clavulate. They are all what? They are all beta-lactamase inhibitors. Is it clear to you? अब फार्माकोलॉजी में ये नहीं बोलोगे ना कौन सा मैकेनिज्म ऑफ एक्शन है? You will be able to understand here. So I hope you are all done. किसी को कोई क्वेश्चन है तो बता सकता है. Then we can say thank you.